Hello everyone, the topic is about blood topic which is related to animal tissues. So blood, the red stream that flows in our body is blood. The red stream that flows in our body is blood, and it is the medium of solids like glucose, liquids like water, and gases like oxygen and carbon dioxide. The wellness or illness of a person identified by blood. While if a person is suffering with any disease, it can be noticed with the help of blood test, right? That is why the wellness or illness of a person can be understood by blood test. It is really amazing that blood is red in color because of the presence of hemoglobin, and we also see blue colored blood in snails because of the presence of hemocyanin. So this contains copper as the element, and here iron. In hemoglobin, heme means iron, and here cyanin because of hemocyanin the blood is blue in color. Here is red in color, here blue in color, and we observe blue colored blood in snails. And of course white color blood or colorless blood in insects like cockroach. We observe colorless blood in insects like cockroach and other insects. Blood contains 60% of water content. It is called as plasma. And the yellow straw colored substance that is obtained after the blood is yellow to clot is called serum. What is serum? After the blood is yellow to clot, whatever the yellow color substance formed, it is called serum. Okay, and there are three types of cells in blood. One is the red blood cells, white blood cells, and third one blood platelets, and we will discuss about each and every cells of these. Okay, next, the cells of blood are called corpuscles. And these are three types. We will mention it in the form of a flow chart: blood cells or corpuscles. Three types: red blood cells, also called erythrocytes. Red blood cells or erythrocytes. Next, this one, white blood cells or leukocytes. And third one, blood platelets or thrombocytes. Okay, and red blood cells, first of all, red blood cells are spherical shaped or round shaped, and some are biconcave in shape, and there is cytoplasm without nucleus. Nucleus is present whenever we are in mother's womb, means embryonic stages. Nucleus is present in our red blood cells. Later it disintegrates. Nucleus disintegrates, and blood is red in color because of the presence of hemoglobin in red blood cells. Because of the presence of hemoglobin in red blood cells, the blood appears red in color. An average adult contains 5 liters of blood. 5 liters of blood. Next, the lifespan of red blood cells is 120 days. The lifespan of red blood cells is 120 days. And generally, new red blood cells are formed from bone marrow of longer bones. New red blood cells are formed from bone marrow of longer bones. And the production of red blood cells is called erythropoiesis. Production of red blood cells is called erythropoiesis. So, how many red blood cells are died? Same day, that much number of red blood cells are produced from the bone marrow of longer bones. And whatever the dead RBC are there, dead RBC, they goes to spleen. They are sent to spleen. That is why spleen is called graveyard of RBC. Spleen is called graveyard of RBC. And what is the function of this hemoglobin? Hem means iron. Globin means protein. Protein. So, and also a substance is present called porphyrin. Porphyrin, an organic substance, is present. Okay. And what is the main function of hemoglobin is it binds, it combines with oxygen and distribute oxygen to different cells of the body. So this is about the red blood cells. Red blood cells. Next, white blood cells. White blood cells are generally their lifespan is 12 to 13 days. And these are classified into two types depending on the presence and absence of granules. Granules are present, they are called granulocytes. If granules are absent, they are called agranulocytes. Okay, and these granulocytes are three. One is the acidophil, basophil, neutrophil. Acidophil, basophil, neutrophil. See, two-lobed nucleus is present, and granules are present, and cytoplasm is present. It is the acidophil. And S-shaped nucleus is present. And it contains granules and cytoplasm. It is the basophil. And third one is the neutrophil, which contains multilobed. More lobes are present, granules are present, and cytoplasm. So this is the neutrophil. Here, what is the function of neutrophil is, neutrophil fight against bacteria that cause diseases. So it fight against harmful bacteria that cause diseases. That is why it is called first line of defense against bacteria, and also called microscopic policemen. Why? Because they are fighting against disease causing bacteria. Next, agranulocytes. Agranulocytes are two types, and granules are absent. Round shaped nucleus is present. It is called lymphocyte. And this is the cytoplasm, and there are no granules. And kidney shaped nucleus is present. It is called monocyte. It is called monocyte, and this is the cytoplasm. Okay, round shaped nucleus is present in lymphocyte, kidney shaped nucleus is present in monocyte. This is about the agranulocytes, where A means absent, granules are absent. What is the function of lymphocyte? Lymphocytes release antibodies and destroy antigens that are entered into the body. Okay, antigens. Those antigens are killed by lymphocytes by producing antibodies. Whereas monocytes move like amoeba. Monocytes move like amoeba and they engulf the disease causing organisms and send the wastes in the form of pus to outside. Okay, so this is about the white blood cells. And blood platelets are thrombocytes. These blood platelets and thrombocytes are also irregular in shape and they contain cytoplasm, but there is no nucleus in thrombocytes or blood platelets. The function is they help in blood clotting. Whenever an injury occurs to our body, the blood will clot within few minutes. It is because of blood platelets or thrombocytes. So this is about the blood platelets. And blood is called fluid connective tissue of the body. Okay, right. Next, blood groups. Carl Landsteiner discovered blood groups. And identified them as A, B, AB and O. Right. We will discuss about each and every blood group. For example, A blood group is there. The persons who are having A blood group, those persons contain antigen A on RBC and they contain antibody B in plasma. These persons can donate blood to A and AB. These persons can receive blood from A and O. Next, B blood group. B blood group persons contain antigen B in RBC, antibody A in plasma, can donate blood to B and AB, can receive from B and O group. Next, AB group. AB blood group persons contain both A antigen and B antigen in RBC and there are no antibodies in plasma. Can donate blood to AB only and they can receive blood from all groups. That is why they are called universal recipients. Next one, O blood group. O blood group contain no antigens and their blood group contains A and B antibodies. Okay, and they can donate to all groups. They can donate to all groups. That is why O blood group is called universal donor. And they can receive from O only. So this is about the blood groups. Next, identification of blood group. Identification of blood group. So how can we identify blood group of a person? Simple. For this we have to take some needles, a slide, wax pencil, okay, anti-A serum 5 ml anti B serum 5 ml, okay, and cotton plug, alcohol or spirit to stop blood flow, alcohol or spirit. First what we have to do is we have to take a slide. On this slide, take a person's drop of blood and place 3 drops of blood like this on the slide. Now place here anti-A serum here, anti-A serum, here anti-B serum, okay, here RH serum, anti-RH serum, this is called anti-RH serum, anti-RH serum. Next, here what we have to do is, here we are taking anti-RH serum, this is the blood, and these are the anti-A serum, anti-B serum, anti-RH serum. Then take a wax pencil and mix this blood sample and anti-A serum, okay, right. If blood clots here, and did not clot here, okay, then the person's blood group is A. So generally mentioned here as A and B. If the blood clots here and here the blood did not clot, so the person's blood group is A. Otherwise, the blood clots here in anti-B serum place, if the blood clots, then the person's blood group is B. If in both cases the blood clots, the person's blood group is AB. Okay. If the blood did not clot in both cases, the blood is not clotted, okay, then the person's blood group is O. Okay. And what about plus minus, means positive, negative? See, it takes some more time for the blood to clot where we added anti-RH serum, If the person's blood sample mixed with anti-RH serum, if it is clotted, that person is said to be RH positive. If it is not clotted, that person's blood does not contain RH factor, so RH negative. So depending on this, we can say whether the person is A positive or A negative, B positive or B negative, AB positive or AB negative, O positive or O negative, like this. Okay, this is how identification of blood group occurs. And we will discuss about another topic in the next class.